Hello everyone. I would like to extend my thanks and appreciation to all organizers of this international conference held in Turkey, fourth International Eurasian Conference on Biological and Chemical Science. My name is Dr. Rasha Karim Mohammed from University of Baghdad, Faculty of Science, Department of Biology, Baghdad, Iraq. My work impact of the saponin of Agave americana leaves on the integrity in the alien tip cells in vivo. My plan, introduction, materials and methods, result and discussion, conclusion. Now, introduction. Iraq, as a tropical country, is endowed with an abundance of renewable resources from the plant kingdom. There are many plants in the kingdom that have varied application, but the humans are unable to use them owing to lack of knowledge. Agave americana is one of the richest sources of potent natural saponin in the plant kingdom, and it has a variety of applications. Americana is a monocotyledon plant that belongs to the Agaveaceae family, which include aloe vera. It normally grows in tropical, subtropical, and temperate regions of the world. The plant-based saponin are triterpenes and steroidal in nature. This figure shows that Agave americana plant in College of Science, Darden. Saponins are a class of secondary plant metabolites that can be found all over the plant kingdom. Saponins are phytochemicals that can be found in a variety of vegetables, legumes, and herbs. Many plant species, both wild and cultivated, have triterpenoid saponin are the most effective in cultivated plants. Although steroidal saponin are widespread in plants used as herbs or for their health promoting characteristic hyaluronic and collagenous. Triterpenoid saponin are a group of compounds found across the plant kingdom that are distinguished by a structure that includes a triterpene or steroidal glycone and one or more sugar chain. The advent of saponins as key commercial compound, along with the physiochemical feature and expanding evidence for their biological activity such as anti-cancer, antifungal, has led to increased demand for food, cosmetics, and pharmaceutical industries. Rapid PCR markers are thought to be beneficial in determining genetic diversity, population, genetic taxonomy, and genetic marker and genetic diagnostic. Using single short and arbitrary oligonucleotide primer, this technique always permits the examination of genomic variation without prior knowledge of DNA sequences. Low-cost development of a high number of DNA markers in a short period of time necessitate a minimal amount of DNA and the ability to produce automorphism. A mutation is a permanent and very unusual change in the number or sequences of nucleotides in genome. Induced mutation and mutation that have been purposely induced by various mutagenic agents. Mutation is a heritable change in an organism that causes various structural changes in DNA, such as change in single nucleotide base of gene, mutation, substitution of one nucleotide base by another, deletion of one or more base pair in DNA sequence, frame shift, mutation, and chromosomal rearrangement, duplication, deletion, chromosome segment. As a result, the project goal were to determine the effect of saponin as genotoxic agent. Materials and methods. Now, this in material and methods, that's beginning with the plant collection from the gardens of University of Baghdad, and then they're dried of leaves and powdered. Then the extraction of crude saponin. Now then, when we extraction the crude saponin, we then studying the effect of the crude saponin on the DNA integrity of onion meristematic cells when growing of the onion on the concentrations of saponins that range from 0, its control, 10, 15, and 20 and 25 milligram per mole for 48 12 hours at 250 to 30 centigrade in dark. And then when we treatment these roots with the sequential situation of saponin, then we will go to genomic DNA and extraction and purification with protocol, and then analysis of PCR products by using rapid primers, estimation of polymorphism. Now results. These results show that this figure show that we use the two primers, OPP11 and OPP03. These primers for rapid primer. Now as we show the number of the monomorphic band and polymorphic and unique band and fragments, as well as the percentage of polymorphism for obtained for LMC treatment. Now this column shows that the fragment size for molecular weight base pair. Now the number of fragments, or there is 30 number of fragments with molecular weights range from 30 to 1070 base pair. Now we show one unique band and 7 monomorphic bands and 4 polymorphic bands with polymorphism with 57.142% of polymorphism. Now this figure shows that DNA samples or onion DNA samples in agarose gel electrophoresis for PCR reaction utilize the random primer OPP11 electrophoresis on a 1.5% agarose gel for 1 hour, 70 volt and 1 XTB buffer. Fractionated the band which were then observed under UV light after staining with helium bromide with ladder 100 base pair. And now this figure, this table that coincides with the figure in the previous slide, that shows this mean band from 300 to 1070 base pair. Now as we see we have 3 hours for treatment and for concentration range from 5, 10, 20, 25 in addition to the control. This column shows that control, or this band, will appear in control, where now 3 band appear in 30, 40, 50 base pair, and in 80 and 90 base pair. Just this band will appear in control when we treat these roots or onion roast tip with the sequential concentration of saponin 5, 10, 24, 25 for 4 hours, we show there is now no any bond appear as control this, and in 8 hours there is no bond appear, but in 12 hours one unique bond will appear in 20 milligram per ml concentration of saponin. This lead that have mutation will appear at this time. Now this band, this mean polymorphic band, appear in 10, 20, 25 for 4 hours, and in 8 hours for 5, 10, 20 milligram per moles. These band not appear in control. The loss of complementary sequences in rapid primer resulted in the absence of the application product in some DNA sample. This means that what happened a mutation. Now this band is appear in all the samples as in control, this monomorphic, and this band will appear in some concentration and disappear in other, this polymorphism or polymorphic bands. Now as we see these 3 bands, the last 3 bands, it's monomorphic band. Now this from this result we show this could be evidence that saponin have ability to cause a mutation in plant cells, that the use of rapid marker analysis to detect DNA polymorphism provide an effective biomarker technique for evaluating the impact of saponins as mutagens. Now conclusions that could be considered that saponins have genotoxic effects when used in high concentration. Now thanks for your listening.